welcome back to Metagenomics Mondays. I hope you can hear me out there. I'm Carolyn Lawrence Dill, and today I'm going to introduce Edo. But before I do that, I want to let you know there are 3 more of these seminars left. July 23rd is Karin Dorman, July 30th is Greg Phillips, and August 6th is Larry Halverson. So please tune in for those Metagenomics Mondays over the course of the next few weeks. So it is my distinct pleasure to introduce Edo Friedberg for a lot of reasons. One is because I get to watch his seminar and I love Edo's seminars. Another reason is because Edo doesn't just work in metagenomics. It seems like anything I ever become interested in, the person that's already working there is Edo. So I think you're going to find that a lot of the things he's working on, he's thought of maybe just a little bit ahead of you, or maybe just a little bit ahead of me. So with that, I will hand it over to Edo Friedberg.
Okay, thank you, Carolyn, for a very kind introduction. I hope I can live up to that. So, hello, everyone. That's my title slide. Well, not quite yet, but you know how it is when you pick up, this is summer, so I decided to give a certain more lightweight seminar, but hopefully you'll learn something. So you know how in summer you hopefully read a little bit more, you grab a book, and suddenly you get this prologue, and you start reading it, and what the heck does this prologue have to do with anything that's in the book? So this is kind of going to happen right now in the talk. So this talk will actually start with a leak of texts between 2 government employees, as is usual these days. And this is Carolyn, who was basically told me that I should give this talk, and I said, well, yes. And then after a while, she started bugging me, you know, you need a title. And yeah, I'll give you a title pretty soon. And eventually she just started texting me, title, please. And I knew I couldn't get away from it. So I said, okay, here's my title. I tried to be incredibly facetious. Maybe she'll let me off the hook. And then I thought, well, maybe not. Let me think about this title. It sounds a little bit too pretentious. She actually liked it. No, I don't really want this. Thanks. I'll think about it, please. No takebacks. Yeah, so that was me after that conversation. Now, not only 2 months ahead am I boxed into giving a talk, I'm boxed into giving a talk with the title, which I really don't know exactly how I'm going to fit it into. So I decided to consult with people about, okay, common perils and pitfalls that we might encounter in metagenomics and in microbiome analysis, and this is how I got the title. And when I started to think about where can I consult with people, you know, I'm kind of isolated here a little bit. Not only am I in Ames, Iowa, but I'm also in the vet school. So this is kind of where really, really hard to find people. So, you know, we can go on social media and so on. And, but I wanted to go somewhere where it's information is reliable and giving in long form, and you know, I can talk to a lot of good people about it, so I couldn't find such a place, so I went on Twitter instead. And I said that I boxed myself into giving a talk entitled, Why Your Microbiome Analysis is Wrong. I need ideas, horror, funny stories. And I tagged a lot of people. Those of you on Twitter may know some of them. And those of you who are not might want to follow them, because a lot of these people are actually very knowledgeable in the field. Adam Filippi, for example, is the guy who wrote Canoe and various other assembler and alignment programs for metagenomics and genomics. Thorsten Seaman wrote Proca, which is the default go-to program for annotated prokaryotes. Mick Watson is a big name in metagenomics in general. Fiona Brinkman, Jonathan Eisen, all these people don't need any introduction, I hope. And if they do, you may want to look them up. So this is actually my acknowledgement slides, and this is a little bit of a community effort. And throughout my talk, you'll see pearls of wisdom in 280 characters or less that are peppered among my different slides from these people. So first, we kind of want to establish a few ground rules, because there's a lot of, you know, when now we read about the microbiome and think about the microbiome, some people think, oh, that's a lump, that's the bacteria that's in your gut or something. And people only think really of the human microbiome or even the human gut microbiome. So let's establish that that is not a or the microbiome. A microbiome is simply the sum total of a microbial ecosystem, okay? So we're talking about, like an ecosystem, we're talking about biotic and abiotic interactions. And the ecosystem can be in or around or about a living host, but it really does not have to be that way. So it's important to establish that from the beginning. By the way, these slides will be available on SlideShare after my talk. And there are a lot of, and I tried to find as many good reading material as I could, and they're all peppered here among the slides. So how do we study the microbiome? And that's another thing that's important. Because we really have two basic methodologies. One is marker gene studies, and another is shotgun metagenomics. People tend to confuse those. A marker gene study is usually for phylogenetic assignment, and that is 16S ribosomal RNA or 18S ribosomal RNA, but it can be any other kind of phylogenetic marker gene. If you're doing a viral microbiome, of course, those are not relevant. If you don't want to, if you want to do some other kind of marker gene study, then, you know, not necessarily a phylogenetic one, or you want to pick another phylogenetic marker, that also works. So these are marker genes, and they only study a single gene only, or rather a distribution of homologs of that genes. Whereas metagenomics is really to extract all DNA and sequence and interpret the lump total of the DNA that you have. So if you're doing marker gene studies, you're not really doing metagenomics, and this is kind of important to know. And going back to Jonathan Eisen's blog, referring to 16S surveys as metagenomics is misleading and annoying. Bad hashtag badomics, hashtag omic mimicry. And he does both, really. Same thing, Mick Watson commented on that. You're probably not doing metagenomics. So let's just take this particular ground rule. One common error that happens to a lot of people with metagenomic study is that they're not really doing metagenomics. So how do we actually do the studies? Well, you know, pick an environment, plant and soil in this case, establish some sort of extraction protocol, amplify, sequence, try to interpret this as best as you can computationally, and publish. So supposedly that will be kind of a simple way to do things. Only each one of these steps and the combination, and any combination of the steps, may have certain perils and pitfalls. I will mostly talk about the perils and pitfalls that occur in the computational interpretation, because that is where I messed up the most. And that is kind of, because I'm a bioinformatician, this is kind of where, you know, my zone of, my area of comfort is at. However, and a lot of you in this room and probably on Zoom also know a lot more than I do about sampling and extraction and so on. However, I will touch upon those a little bit as well. I'll start from extraction because it's kind of interesting. This was information that was given to me by Mick Watson. So this is an old paper from 2004. Sorry, you can't see. Oh, you can't see the dates. Okay. And in this paper from 2004, you know, it was even, you know, really there was no real metagenomics at the time. There was barely any 454 sequencing, but microbial ecology does predate metagenomics. And in this paper, the impact of DNA extraction method on bacterial community composition, you know, can find some very sort of worrying quotes. We believe that the choice of DNA extraction protocol will influence not only the determined phylogenetic diversity of indigenous microbial communities, but also the obtained functional diversity. So we're looking both at 16S and whole genomes, whole metagenomes being you know, disturbed by your choice of extraction protocol. And we find that the choice of DNA extraction protocol had significant influence on the fingerprints of the bacterial community. But, you know, how bad is this really? So this is another study where the researchers looked at the choice of bacterial DNA extraction method from fecal material. And this is a bit later. This is actually a proper shotgun study. And the curves that you see here are rarefaction curves. Rarefaction curves show you basically the diversity of whatever you are looking at in your samples. And if our rarefaction curves go just straight up, that means there's a huge diversity, and actually you're not sampling enough. Once our rarefaction curve starts to plateau, like you see here, that tells you that you are entering the mode where you're resampling all the time and not discovering new things. So that means you are sampling at almost a correct rate. So this is a very common ecological tool and microbial ecology tool as well. And here they're sampling not for species, they're sampling for different numbers of genes. And each, we're looking here at 3 individuals, okay? So we see individual B, individual A, and individual C. And individual B is all these different blue color codings. Individual C is magenta, individual A is sort of greenish yellow. And these are basically different extraction methods taken from the same sample. So they took the same samples from individual B, did different extraction methods, and you can see the variance in the results that they got based on their rarefaction curve. So the rarefaction curves do cluster by the individuals, but there is still a substantial difference between the different rarefaction curve depending on the DNA extraction method. So maybe things are not all that bad. We're getting some sort of, you might call it an error variance or protocol dependent variance, but at least if we study this by the metric of rarefaction curves, the same individuals tend to cluster together. Okay, so that's good. However, then again, we might get some things that are kind of messed up. And this is from another study that was done really at 3 different locations. And here they looked at something else. They took the exact same samples and they shipped them to 3 different institutes. ICL is Imperial College London, UB is University of Birmingham, and WTSI is a Wellcome Trust Sanger Institute. So all over England. And they basically followed the exact same protocol in those 3 institutes. And this is for really extraction and amplification only. So you got serial dilutions, and those serial dilutions were amplified. And on the Y, and so this is on the X-axis, number of serial dilutions they did. And on the Y-axis, the proportion of total sequences as they were assigned to different species, okay? And you can see the consensus bar on the right, marked by CON. So as you can see, working in 3 different labs with the exact same protocol gave them really 3 different results in the amplification stage. So this is like the qPCR that they did. So things might actually quite be bad, and this kind of tells you something about the problems of reproducibility that we encounter in this field. And like I said, I'll get pearls of wisdom here from various people. So this is Adam Cribbs, and this is something he never actually published for obvious reasons. He said that using a human single-cell SMART sequence experiment turned out that the enzyme was contaminated with bacteria, and he could detect about over 100 different species. So he did a single-cell experiment in human, and he found 100 different bacterial species that weren't really there in his culture. They were in what is subsequently known as the kitome, the microbiome of the kit. Wonder how much this actually happens in microbiome analysis without anyone noticing, right? Because when you do this in human cells, you immediately notice that you have a contamination. If you do it in a microbiome, not so much. So that is a problem, although not necessarily, because one reply to this tweet was, your failed experiment is another person's Nature paper. So wonder how many of these papers actually show contamination versus not. So that was a little bit of my talk on some aspect of the actual experimental or wet lab aspects. Let's talk a little bit more about computational issues that might arise. Just to look at the protocol of how we analyze microbial diversity. So, you know, you would have your marker genes, and with those you'll do taxonomic profiling. And then you can go into higher level analysis, such as abundance and diversity. Or you can do full shotgun metagenomics. And then from full shotgun metagenomics, excuse me, You can also do either taxonomic profiling by basically binning those into basically binning the reads that you have, not only marker genes, and we'll get into binning in a minute, and functional profiling, which basically tells you what they're doing. So marker genes tell you who's there, and functional profiling tells you what they're doing. Usually there's this tendency to do some sequence assembly. So we all know about sequence assembly that we do just prior to well, when you actually do a whole genome, right? But you can also do sequence assembly with metagenomes. So I'd like to, some of you bioinformaticians here probably know about sequence assembly, so excuse me. I will talk a little bit about the nuts and bolts about sequence assembly and how this can affect, how these nuts and bolts can affect the way you do your metagenome analysis if you do assembly. And if you do 16S, you probably do assembly anyway, because you need to put in all the reads together to reach your V1 or V2. If you do shotgun metagenomics, there are really two schools of thought. Should I assemble or should I not assemble? I assume here there's assembly going on, and you might want to understand what's going on in assembly because that really impacts, as I will show you, as to the results that you might get. So how did sequence assembly start? To do that, we need to understand a little bit of the math behind it. This is a map of Königsberg in 17th century Germany. And the good citizens of Königsberg had this little kind of game they had where they tried to cross the bridges, marked here in blue, between the different areas on the river that flows through the city, and try to cross each bridge only once and end up in their starting position. It sounds like a great kind of pastime. The only problem is nobody actually managed to solve this problem until along came Leonhard Euler, a Swiss mathematician and guy with a horrible sense for headdresses, that decided to, he was a mathematician, and as a mathematician he said, well, I can actually represent this problem in a simpler manner that will hopefully solve it. And thus Euler invented graph theory. So now each island or landmass in the city is represented by a colored dot, and the bridges are represented by lines connected these colored dots. And if there are more than one bridge between two colored dots, like between the red and the green, you get two lines. The lines are called edges, and the dots are called nodes or vertices. And Leonhard Euler built a whole theory that is studied to this very day around this idea of vertices and edges and connecting graphs. So this is really a graph. And a graph is called Eulerian if each edge can be visited only once. Euler actually managed to prove very easily that solving the problem of the Königsberg bridges is impossible, which is why they built yet, I think that's why they built yet another bridge just to be able to walk across all the bridges once and visit everyone. So this is an Eulerian graph. Then if you actually like to go within certain directions to visit the bridges, then you can have a directed graph. So each edge like this has an arrow on it, and then the arrows you can walk between the bridges and say, I need to walk only on this way or that way. Think of the bridges being like one-way streets. And each node has been defined. It can have in degrees and out degrees, meaning you walk into the landmass and you can walk out either on two bridges or one, and so on. Finally, a directed connected graph is called an Eulerian graph if it has mostly two semi-balanced nodes, meaning a node with one input and two outputs, or two inputs and one output, and so on. And it is mostly balanced in the other ways. What has that got to do with sequence assembly in the first place? Well, actually sequence assembly takes place on an Eulerian graph, as I can show you, and I'll show you in a minute. And now that we know that basically we need to walk between segments, this is how it's being done. So here we have in A, our very simple, very small genome. And let's do some short read sequencing. Actually, by the size of the genome, even short read sequencing will be pretty large size compared to the size of the genome. So we sequence this genome and get these various reads that you see in B, CGT, CAA, and TGC, AATG, and they have these overlaps. And you can use these overlaps to draw edges between the different reads in your genome. Once you draw the edges, okay, you can have an overlap, as you see on, is that right? Yeah, right. On the right side, and you can reassemble your genome from the read. Remember, you don't have the whole genome, you only have reads and you can assemble them. That actually works well if you have small genomes and one read. However, if you have many, many genomes, like you have in a metagenome, and you have millions of reads, you may want to consider a slightly different strategy, or a really different strategy, in which you assign k-mers. K-mers are basically, they're not reads. K-mers are basically, after you do your reads, you decide to break up these reads computationally, not for real, computationally, into lengths of 3 or 4 or 5 or 20 or whatever. So you assign these k-mers, and you can see here that we did a k-mers of length 3. Each k-mer is in a vertex, and ATG is overlapping by 2 with TGG, which is I'm reading clockwise from 12 o'clock, which overlaps with GGC, which overlaps with CGC, and so on. And then you try to solve this by walking through each vertex once. Okay, that is one way of assembling your sequence, which is great. However, this is kind of a hard problem to solve. It's also called the Hamiltonian cycle. So in 2001, Pavel Pevsner came up with the idea, let's try to solve this like Euler did with the bridges of Königsberg, and do an Eulerian cycle visiting each vertex once. So the k-mers are now on the vertices, and the k-mers are now on the edges by visiting each edge once. That's what I meant, each edge once. The k-mers are now on the edges, and the vertices are the overlap, so to speak, or they're really the k-mer n-1. So AAT, as you can see, is flanked by the node of AA and AT, and ATG is flanked by the node of AT and TG. So now we go through these, and we solve the assembly problem as an Eulerian cycle. And if you have an Eulerian cycle, you manage to assemble your entire genome. If you don't, which usually is what happens, because even with a whole genome, not necessarily a metagenome, you do get these, these contigs. That basically means you couldn't do an Eulerian cycle for all your k-mers, so you broke them up into these clusters and assemble as many Eulerian cycles as you can, and those are your contiguous segments that you get in genome assembly or contigs. I hope you understand that the k-mer is a really very, very important parameter when you do your assembly, and picking your k-mer during your assembly stage is of paramount importance towards the quality and comprehensiveness of your assembly. And this is a whole bunch of different assemblers that are going on here. And as you can see, most of them, if you can see the method column, if you can't, I'll read it out to you. Most of them, except for one here, which you really want to see what exactly it does, all use De Broglie graphs and with single k-mer or multiple k-mer lengths. So it's really important to understand they all do more or less the same thing, and the differences lie in the details of how they assign k-mers and various other tips and tricks to make them a little bit more accurate and so on. But they're not all the same method. If we look at performance, the performance really, really differs, as we can see from this particular, from this study here. And this study, the researchers took 2 microbiomes, one from kelp and one from the forest soil in Marburg. So the kelp is kind of a low complexity microbiome. It doesn't have too many associated microorganisms, whereas the forest soil does have a lot of associated microorganisms. And on the y-axis, you see various metrics that are used to understand the quality and comprehensiveness of the assembly. And on the x-axis, or basically the different colors, are associated with different assemblers that are available for anyone to use. So for example, N50, which you see, which is second from top, is you might think of it as kind of not exactly, and those of you who know N50 will know that what I'm saying here is not exactly true, but it's kind of the median scaffold length that you manage to assemble. The third from the top is how many scaffold numbers you have. You want to actually minimize that. N50, you want to maximize that. Scaffold length, you want to maximize, and so on. So you see these assemblers vary hugely in their performance. So it's important to see that. No, somebody's happy with my talk. All right. Then we also look at the cost efficiency, because it's also important. You don't want to assemble your genome forever. So if we look at cost efficiency in terms of CPU time or performance, which is kind of, I don't know, they kind of did their own metric for performance here, which is the mapping rate times N50. So kind of a speed versus comprehensiveness. You can see that again, they vary quite a bit, the assemblers. So that's an important point if you're assembling your metagenome or microbiome, as many people do. Because these methods perform all differently, and each researcher touts their own method as the best there is out there. A group of researchers in Germany again, led by Alex Scherba, decided to hold a critical assessment of metagenome interpretation, or CAMI, where they basically gave out various synthetic and true metagenomes and asked people to assemble them and see how that works. They actually tried assembly, binning, and taxonomic profiling. I haven't gotten to binning and taxonomic profiling yet, but I would. But now we're talking about the assembly challenge. And the idea here is that you take a lot of researchers that say, hey, you contact a lot of researchers that write these assemblers, and tell them, hey, can you please take this microbiome? We know what it should be assembled like. You don't, and it's a synthetic microbiome, so they kind of gave them, they kind of hid the true answer. And please run it. And they had 16 teams that worked in their first challenge. It submitted altogether 25 programs, 215 submissions. I won't get into the differences here. And they, for the bioinformaticians here, this is actually very good practice. The organizers of CAMI actually required the software to be run in a standard fashion on something called a Docker BioBox. So it can really be run on a virtual machine. Docker is kind of a virtual machine, can run on a virtual machine anywhere. So that helps replicate the results and assign replicability. And these are the results from the CAMI Assembly challenge. Each color is a different assembler. So you have certain assemblers that are running in with different k-mer parameters. So you can see velour K63 or velour K31. K31 means k-mer of 31. Velour 63 means a k-mer length of 33 and of 63 and so on. And they ran them in different parameters with the challenge microbiome. And the fraction of the metagenome of the genome that was assembled, remember there are several genomes, that's why you have a distribution. Some genomes were assembled well, some genomes were not assembled well. So the median, these are box plots, the median is the line right there in the middle, and you have the upper 25% and lower 25% right there. As you can see, the genome fraction, that there's a huge difference again in the performance of the different assemblers. A is for all of them. B is for only those genomes that have 95% identity or more, so you might call it the easier challenge. And C is for those genomes that have a 95% identity or less, so kind of a more challenge and a more kind of diverse metagenome. And you can see that in C, the performance is not as good, and B, the performance is fairly good, especially for megahit, and in A, it's kind of all over the place. So really big differences in assembly performance. And it's important to remember, as Murat Eren from the University of Chicago says, that k-mers aren't reads, reads aren't genes, genes aren't genomes, and genomes you get from metagenomes are often nothing but consensus sequences of large population of cells heterogeneity, which is defined by heuristics of your assembler, which you likely know next to nothing. Okay, that's depressing. But you have to keep that in mind, okay? You have to keep in mind that you actually have an ecosystem of fragmented genes that you're trying to interpret. So that's it about assembly and so on. Now we're going to move into taxonomic classification, which might be of even more interest, hopefully, to many of you. And that is basically taking your metagenome and trying to assign it to different, well, taxa, right? So again, there's a plethora of different types of methods that are used to assign metagenomes to different taxa. They can be either alignment-based, which means you're basically using something like BLAST or like Diamond, and then doing some hacks over that to get to understand what's going on. So you would have a reference database with all possible, with all our known taxa there, and you find a read, you blast that read against a certain taxon, and you blast that read against your database, you find the closest taxon. Oh, this is not as simple as it sounds, and assign that read, and then you kind of do and repeat for all the reads that you have a few million times, and in the end you end up with some sort of distribution based on the alignment. Composition-based is a little bit more sophisticated, and that helps you do some de novo assignments as well. Composition-based means you're not doing alignment, you're looking at the nucleotide composition of your metagenome. As we know, different taxa will have different characteristic nucleotide composition. And if you can guess your nucleotide composition, if you can predict your genome from nucleotide composition, you're golden. And these are different methods that do that. Many of them do hybrid as well. So they do both assignment by alignment and assignment by composition. And actually, there are quite a few more hybrid method than here. It's quite a few of the composition methods that are shown here are actually hybrid method, but with an emphasis on composition. I won't get into the assignment independent today. However, even assigning simply by alignment is not that easy. I want to show you here a very simple example of a method called Megan. Megan is really a very good method. You'll see its performance later for taxonomic assignment. And this is Megan 1. I'm showing you Megan 1 instead of Megan 5 or 6, which you have now, just to illustrate the simple example. What the Megan people decided to do is very simple. You take a query and you blast it, and you find all the top hits that are scored by some sort of bit score. And so we all have the same query 79 to 2. It's all the same query, and you're just getting different blast hits with different scores, but they're all significant. And you find a hit in Vollinella, and you found a hit in Helicobacter hepaticus, and you find a hit in Campylobacter. All right, great. So where did this fragment come from? Where does it actually belong? Well, we don't know. But what we can know is that it probably belongs to the most recent common ancestor phylogenetically of all 3 of these taxa that you hit. So you assign it using the lowest common ancestor, LCA, to Campylobacterialis. So that is really all you can know, and that is how Megan does the assignment using the LCA. So Megan, I believe, was the first method to use this LCA assignment. So on the one hand, so it's kind of being very, you might say, almost under-predicting or very cautious with its prediction. On the other hand, KRAKEN uses LCA, but in a different manner. It uses exact k-mer matching. So this is your query sequence. It breaks it up into small k-mer, not to be confused with assembly k-mer. This is a totally different use of k-mer. And it goes for exact matches. The nice thing about doing exact matches instead of alignment with gaps and so on, it's really, really fast. So KRAKEN is an incredibly fast method. And that actually is quite a bit. You know, you might want to, in many cases, you do want to trade off fast method for some of your accuracy. And KRAKEN does trade this off a little bit. So here we have all our different k-mers, and they could be assigned, the blue ones can be assigned by LCA to the blue node of the phylogenetic tree. There's one purple one assigned to the purple node. There's one red one that might actually be an error, was assigned to the red node, which is way off somewhere. And then we have a couple assigned to the orange node, which is way on the bottom. KRAKEN overpredicts. I hope Steven Salzberg is not listening to me here. He's the guy who wrote it. But KRAKEN does have a tendency to overpredict, and that should be kept in mind, because given this particular example that I have here, KRAKEN will say your particular query sequence belongs to 4. And that might actually be a problem. Why? Let's look at comparing different binning methods. What happens if you take a metagenome and you try to bin all the reads in the metagenome, do taxonomic assignment, but you remove one particular taxon from your entire reference database? This is called the Clade exclusion. This is called Clade exclusion. You remove a whole clade from your database, and you try to see, and you try to sort of see what does the binner do? What does your phylogenetic assigner do? And that's exactly what Fiona Brinkman and her group did when they tried to compare these different metagenomic assigners. And if you do assignment by clade exclusion, that means that, for example, if you remove an entire clade from your metagenome and you try to assign something that's in that clade, your binner will give you really either 2 answers. Either say, I don't know, it's not there, which is probably a good thing to say, or it'll try to find the closest whatever it can find, even if it's completely wrong. And sometimes that actually happens. So as you can see, by clade exclusion assignment, KRAKEN is not a very good performer. You see the sensitivity on top and the precision on bottom. KRAKEN has a problem there. And note that they took out species level, genus level, family level, order level, and class level. So KRAKEN tends to overpredict, but by this particular metric, it does not perform well. However, in the wild, as we say, it actually performs well, because clade exclusion really penalizes overprediction very, very harshly. But when KRAKEN was participating in the CAMI challenge, now the CAMI challenge is in binning, not in assembly, when KRAKEN was participating in the CAMI challenge, we see that it is really performing very well. The green line is the average precision. The blue area is, sorry, the green is the average recall. The blue is the average precision. You get the general accuracy in the magenta line on top. And the misclassification error is the orange line. And again, CAMI also tested how well the assignment does in different levels of taxonomy. So everybody did very well in super kingdom assignment, phylum, class, order. Once you get to species, once you get to family and genus, and definitely species, the ability to predict really, really drops up very, very quickly. However, KRAKEN did well in this particular kind of field assignment. So this is kind of an example of different metrics giving you completely different assessments of reports, but you have to know what your question is. If for example, if your scientific question in your microbiome will be sensitive to overprediction, you might not want to use a method like KRAKEN. That will be usually in kind of low abundance, low number of species, a microbiome where you really want a higher precision, and you really want a don't know answer over a maybe answer. But in high diversity or very rich microbiomes, yeah, that works well. Adam Filippi chimes in with this particular nugget. If your metagenome has monkey DNA in it, classifier might call chlamydia present in your sample because some chlamydia genome assemblies were contaminated by monkey DNA from cell culture, and other variation on that theme, which brings us back to contamination. Contamination does not just happen, wet contamination, your kit is contaminated, your sample is contaminated, your lab is contaminated, your database can be also contaminated, your reference database is also contaminated. And we know about microbial contaminations in the human genome, quite a few of them actually. If your metagenome contains eukaryotic DNA and your k-mer-based classifier doesn't know about those eukaryotes, it will predict all the things, the common problem that we have with overprediction. So take care to see that. All right, so now everybody's pretty depressed. Oh, you know, I have to download these assemblers and run them, and I don't know which assembler I want to run, and the binning part is really problematic because if who overpredicts and who underpredicts, and what do I do? Servers to the rescue. They're actually web servers. You can upload your entire metagenome to a web server, and these web servers have the data, and they have the software, and they have the CPUs needed to do that, because you know all these analyses really can't be done in your laptop, except for the software that Xiaohu, who's here in the audience, is developing in my lab, and we already have a really good classifier that can run on your 4 GB laptop. And if you have a 4 GB laptop, please buy a new one. And they give you pie charts, which everybody loves. Okay, so we have all these different web servers that can do the job for you. They already have the pipelines ready in place. You really only have to upload your data and trust the software. Oops. And hopefully get those really nice pie charts and bar graphs that you would have. Okay, great. Except for a certain problem that I personally encountered and like to warn you about. If you decide to do your analysis on a web server. So setting aside everything else, that you actually trust the pipeline and think that the people in the web server did the best choice. We are really just departed from the Friday the 13th. So I decided to do this little poster here talking about a personal experience of mine, which I will call the web server data massacre. And that was a pretty horrifying experience, but like the 2 remaining people from the chainsaw massacre, I actually managed to somehow survive it, as did my data. So what happened here exactly? Well, again, this is a bunch of emails that were in a server in my closet, but I couldn't find them, so I had some colleagues from Russia to send them to me, and I got them. And this is basically, this is actually a real exchange that I had with some people that are responsible for a certain server. They actually were great. I mean, I think that server is fantastic. I won't name it here because this is a kind of a recorded thing and might say as if I'm dissing them. I'm not. I think they responded very well, and I think they managed to save my data and everything, but it's kind of a warning both to people who consume web service software for doing their analyses, and for you who set up your own web servers, as a cautionary tale. So one day, it was actually March 2nd, 2011, I received this email from the mailing list of that server. You're receiving this email because you're a user of our system and a member of our users mailing list. We are happy to announce the release of something Cloud, version 3 of the metagenome analysis server. Find the detail about our new server here and there. Please note, we have disabled certain features in version 2 in preparation for the update. I was working to that minute on version 2 for analysis of my metagenome, and suddenly they disabled it. Wow. All my data were in version 2. Suddenly there's a version 3, and it works completely differently. Yep, my data got massacred. So I immediately emailed them, hi, congratulations on the migration to version 3. There was obviously a lot of work put into this effort. That was the nicest thing I can say. You can see I didn't say there was a lot of thought put into this effort. However, we are concerned about the abruptness in which the migration was performed and ensuing disappearance of our data. We're in the middle of analyzing these data based on the old pipeline, and now it appears 2 months of work may have been lost. Even if the new server is better, we still require, for reasons of provenance and reproducibility, results from the old analysis pipeline. Furthermore, at the moment, we are currently in the process of submitting an abstract to ISMB. That is a conference we were going to, and this data occlusion jeopardizes our submission. Please, we need our old data. I'm crying here. They responded in like 3 hours. Hi, Ido, version 2.0 is available at, and they gave me a backdoor URL. For the near future, we will let our users know before we pull the plug. Thank you, guys. Sorry if you did not communicate this effectively. Whew. All right, data saved. And that was for a conference. Well, about a year later, you know, basically we worked on everything and we were really ready to publish. This is actually the paper that we published, a metagenomic study of diet-dependent interaction, gut microbiota and host infants. So if you really want to know what server it is, you have to read my paper. It's all there. And in this paper, we did some of our analysis on that server, quite a hefty bit, and I decided we actually have a systemic problem here. And so I sent this to the general user discussion mailing list. I hope my email reaches the users of this particular server because I'm interested to know whether there are others who share my problem. But at the very least, maybe the developers can look at it. May they live long and prosper. My questions regard the version 2. By now, we are now publishing research acquired using version 2. I pulled down these data to my disk when I realized the version 2 was going out. But in the interest of maintaining a history of data analysis, would it not be prudent to maintain a read-only versions? Otherwise, if we lose data in each upgrade, or rather the data gets reanalyzed using new tools and new versions, it would be impossible to use the analysis confidently. There'll be no persistence. Another options would be an archiving tool. I had 2 students that worked for a week just pulling down all the V2 analyzed data. So any thoughts and comments, best Ido? Again, they replied very, very quickly and took my problem very seriously. Hey, Ido, the issue we're pointing to is very important. And the comments are good, but solving this is a resource problem. I can't argue with that. We've never simply been resourced to worry about this kind of thing. We have ideas, but you might as well use wget or curl. These are common web-based tools to get stuff off the web and in batch. And that's actually what we did. So this is a call to the funders. Please, when you fund a server, please take into account requests to, for some persistence. That is actually very important. Thank you. And maybe there should be a code of conduct for predictors. Based on everything I said up till now, never force predictions. Always have an unknown category, good for your assemblers. Provide free software, free as in speech, right? You can sell it, but make it free to run and to modify. That ensures reproducibility. Some of these servers give their software completely for free, some don't. Provide access to old versions, very important. Important for user analysis continuity. And if you're running a web server, please archive software and data. Thank you, Fiona Brinkman, for these insights. So after I managed to depress everybody, and oh God, what are we going to do now? Is there a best practices thing? Yes, there are. There are a couple of really good best practices paper that I recommend reading. One of them by Mick Watson's group and Applied in Environmental Microbiology talks specifically about the madness of the microbiome, attempting to find consensus best practice for 16S microbiome studies. He doesn't, spoiler, he doesn't find a consensus best practice because a consensus best practice is non-existent. It really depends on your scientific questions. However, he does delve very, very thoroughly into the various considerations that you need to do with a 16S study and gives a lot of good reading material there, a lot of good advice. Another from Rob Knight's group talks about best practices for analyzing microbiomes. This is a more general thing, not only for 16S microbiome studies. And they talk about some, quite a few caveats there. And maybe, and I'm entering here the final stretch now, and that's funny because I'm entering the final stretch, but it seems like I'm just beginning because I'm putting here a flow chart of how to do best practices in each step. These are the steps you need to look at and consider when you do a microbiome study. What is your question? First and foremost, you always need to know a question or a bunch of questions. You can do a fishing expedition, but even in fishing expeditions, you kind of need to know whether you're going to go fishing in Alaska or in the Caribbean, and you know what kind of fish you expect to get, and so on and so forth. So you need a good study design. Once you design your study, you need to do your power calculation. determine what kind of metadata to collect. That's very important because at some point you associate the microbiome that you have with the metadata that you have. And you want to know the age, if it's an animal, say an animal study, you want to know the age of your animal and what it was fed and where it was kept and the temperature. And all of these, every single thing might be a factor in affecting what your microbiome might look like. Dysbiosis, all of these. Again, you want to do a high-level community profiling. You want to do an actual functional profiling. You want to do a real-time functional profiling. And real-time functional profiling means metatranscriptomics. Not getting into that. That's a whole different thing. No time to talk about that. We did talk about beta-genomics and marker gene studies. You would not, there are various, filters that you'd like to go through. I didn't talk about them here, but you want to know that they exist. You can't just take your metagenomic data. You want to remove various contaminations from it. Here we have also read-based profiling, KRAKEN, Megan, and human N. By the way, human N is not a read-based profiling method. It assigns specifically to the human microbiome. And various high-level analyses that you may want to do. I'm sorry, but the bottom part of my slides are cut off because of the share, but of the Zoom share. But all these slides will go up on SlideShare pretty soon. So I'll send out the slides to anybody I'll send out a URL to the slides to anybody who's interested in seeing them. We talked about best practices in amplification. DNA sequences vary in these primer amplified regions. So that's important actually in 16S, okay? If you're doing amplification, tune your primers correctly. Some DNA sequences amplify some primers amplify certain DNA sequences more than others. This creates huge biases, especially if you're doing an abundance study. Low biomass samples are particularly susceptible to that. That's one point to think about. Binning and taxonomic profiling, we went through this quite a bit. Binning is usually done vis-a-vis some sort of database. So your database choice is crucial. You want a well-characterized environment. Use specialized databases. That will, for one thing, decrease computational time. So if you're doing a human gut microbiome study, you don't want to do the whole soil, or you don't want to do a soil data set, right? And there are various specific data sets for well-studied environments, like the human gut, like the ocean, and various others. If you're using a poorly characterized environment, which nobody knows anything about, well, you just need a database that covers as much as you can. Different types of assignment methods. Again, marker-based methods such as Metaflan will give you a quick, precise analysis. So this is kind of an alignment method. If you want a broader view, okay, use a less characterized microbiome. You can use something like Discriminate for your taxonomic profiling. Megan, which I've shown you before, the one that does the LCA, and Megan 5 is much more sophisticated and accurate than the original Megan, is a really nice one that I recommend if you're just starting out on taxonomic profiling, simply because it's so easy to use, it gives you such a nice visualization, really user-friendly, and it is quite accurate. Functional profiling, again, the database of choice is really important. You can use reference databases for better characterized microbiomes. If you have a bacteriophage microbiome, use FASTER, or at least for starters. ResFAMs for antibiotic-resistant genes, important to a lot of people working here at this university. FOAM for environmental samples, TARA for ocean samples. And again, human N for human microbiome functional profiling, RefSeq and PFAM if you just can't avoid using general purpose functional profiling. And I guess I'm done. So I'll take any questions you may have time to ask. Thank you very much. And I guess people will also be asking questions on the Zoom chat. So, oh, there's already a comment here. Public service announcement from John Shea. The BLAST step will kill you computationally for Megan. Please use a more efficient alignment algorithms. Yeah, that's right. So you can use Diamond. Diamond is kind of the new BLAST, something that is much faster than BLAST. That's a good point, John. Thanks. Any questions? All right, guess not.
I have a quick question.
Yeah.
Tell my students to write down what they did, not why they did it.
Yeah.
So, do you see in microbiome analyses people actually explain why they made the choices they did ever?
In a lot of microbiome papers, yes, they do. Usually it's a little bit in response to the referees, I think, because that's what I did at least, so I don't know, that's an N of 1. But yeah, a lot of people explain we used this binner instead of that, we used this binner because we worked with this kind of data set, with this kind of abundance and this kind of reference and so on. Oh, sorry, I will have to repeat the question. The question was, so Carolyn just asked a question, whether people not just write what they did, but also why they did it. And my answer, in many microbiome studies, I see that because they kind of need to justify why they use a certain type of software and database that are supposed to work well with their data as opposed to others. Any other questions?
Right.
Okay, thank you very much.
Thank you, Lydia.